Alright guys, welcome back. Today we will finish up the chapter on the cell. So we're going to keep talking about membrane transport, right? Just how things get in and out of the cell. We talked about diffusion and osmosis, right? When things go from high concentration to low concentration. Now we're going to talk more about carrier-mediated transport. Carrier-mediated transport is when there's a special protein that grabs onto something and transports it across the plasma membrane. Now, these carrier proteins stretch completely across the plasma membrane. They stretch completely across the plasma membrane, forming little channels or pores. And because of that, they allow something to cross the membrane without ever coming in contact with the lipids in the middle. These carrier-mediated transporters, these channels, are opened by, in multiple different ways. They can be opened when something binds. So for example, like a neurotransmitter can bind to the channel and that can open it. They can also be opened by actual physical distortion. What that means is like squishing of the cell. So our touch receptors, for example, that give us the sensation of touch, pressure, those are actually opened by literally squishing them or pushing them opens a channel. And then some channels are opened by a change in electrical charge. Remember I told you guys before that inside the cell is negative compared to outside the cell. Well, there are some channels that open when we change that charge. Overall, though, we have these proteins that stretch across the plasma membrane so that something can get in or out of the cell without coming in contact with the lipids that are in the middle of the membrane. These transporters exhibit specificity, right? Like specific. That means that one transport protein only works for specific substrates. So it's not like a big channel opens and everything can just rush into the cell or rush out of the cell. This channel will open and it is very specific about what it's going to allow to move through it. So like we have a channel for sodium. We have a channel for calcium. We have a channel for chloride, right? They're very specific in what they allow through. This is just kind of a generic example of a carrier protein, so you can just see the overall point of what it's doing. If you look at the picture, this is the extracellular fluid. So that means that this is showing us what is outside the cell. And then this over here is the cytoplasm. So that means that this is inside the cell. You'll notice this is the plasma membrane, right? Our phospholipid bilayer. And remember that the whole center area, right? All of this area where these little tails are, that's all really, really fatty. So anything that likes water is not gonna like that fatty area in the middle and it won't come in contact with it. So if I have something out here like say glucose, and I wanna get glucose into my cell, the glucose is repelled by these fats. It doesn't like them. So it will not just go straight through. So we need another way. Well, we can have this protein. And notice that the protein completely spans that whole membrane so that when it's open, the glucose can go into the cell without ever touching these lipids. But you'll notice when you look at this that it's closed, right? Look at this. The glucose can kind of come into this channel. The glucose can come into this channel, but it's closed right here. So it has this specific receptor site, this little binding site. And if the glucose can fit perfectly into this binding site, because remember it's specific, so if it doesn't fit perfectly, it's not going to work. It has to fit just right, like a key going into a lock. So because this glucose fits perfectly in this receptor site, if you look at the second picture, this is what happens. The whole thing changes shape. And when it changes shape, the glucose is released inside the cell. You will notice that when the glucose is being released into the cell, look, this side of it, closes, right? So it's like it's either closed on this side or closed on this side. And that's how it prevents random stuff from just rushing into the cell, right? It doesn't just like open up and allow a bunch of stuff through. This is how it's specific. The molecule that's going to go through binds to its receptor site. If it matches, then the receptor will flip and push it into the cell. When we look at carrier-mediated transport, carrier proteins can transport ions into and out of the cell. They can transport organic substrates, so these are things like glucose, for example. Sorry, my pen's not writing. Okay, well, oh well. They can transport organic substrates like glucose across the plasma membrane. There we go. And there are multiple different types of carrier-mediated transport. So when the cell is moving ions or glucose or amino acids across the membrane, there are a couple of different types of this. There's facilitated diffusion, which is really simple diffusion that we've already talked about, right? We already said that diffusion is when things go from high concentration to low concentration. Now, facilitated diffusion is still going from high to low. It's just that the molecule that's diffusing, it can't go through the plasma membrane. So you need some sort of a little channel there. So for example, if I have my cell, right, and my plasma membrane, and I have, say, a little channel here for sodium, right? And it's closed normally, but that's my little channel for sodium. Remember, I normally have much more sodium outside the cell than inside. So my cell looks like this. Now, diffusion tells us that this is going to go into the cell, right? It's going to go from high to low, so from outside to inside. But remember, this sodium can't diffuse straight through the plasma membrane because it's charged, right? It doesn't like the lipids, so it can't do that. Instead, what happens is if we go ahead and open up this gate, right, so that the channel is open, then the sodium can diffuse through the channel. Okay, so it's facilitated diffusion. It's still diffusion because it's going from high concentration to low concentration, and it is passive. Passive means that there is no ATP required for this to happen. The cell does not have to spend energy for this to happen. All it has to do is open the door. And once this door is opened, that sodium is going to diffuse into the cell. So it's still passive, it's still diffusion. We just call it facilitated because we have to facilitate the process. We have to help it. Since the sodium won't go straight through the plasma membrane, we have to give it a pathway, right? That protein, the channel, is the pathway. So it's diffusion that we have to help by giving a channel or facilitated diffusion. Active transport now is still moving things across the plasma membrane with a protein, but in this case, it is active. This is not just passive going down the concentration gradient. This is an active process that the cell burns fuel to do. It doesn't naturally happen. So the cell has to use ATP, right? Remember, ATP is like the cellular fuel. The cell has to use that fuel in order to move these things across the plasma membrane. The reason it has to use fuel to do this is because it's not always going with the concentration gradient. It's typically actually going against the gradient. So it's typically going up the concentration gradient. What we mean by that is it's moving things from low concentration to high concentration. The way that I always think about this is like, think about if you have a hill, right? So up here, this is up high on top of the hill. Down here, this is down low at the bottom of the hill. Now think about it, if you have a ball and you set the ball right here at the top of the hill and you just let it sit there. You don't kick it, you don't push it, you don't throw it, you just set it there. Naturally, the ball will roll down, right? You don't have to spend any energy. Because of the natural forces of gravity, that ball will roll downhill. Okay, diffusion is the same way. Diffusion will go from high to low naturally without any energy having to be spent. But the opposite is not true. Okay, again, think about this hill, and if you took a ball and you set the ball here at the bottom of the hill and you just set it there, is it going to roll up the hill? No, it will not naturally go up the hill. Things do not naturally go from low to high. You have to put energy into it. So you can make this ball go up the hill, but you have to push it, carry it, kick it, throw it. You have to put energy in to make it go up. Okay, same thing with the concentrations. If you want it to go up the concentration gradient from a low concentration to a high concentration, you have to use energy. Okay, so the cell has to use ATP, or cellular fuel, to move things up the concentration gradient. So with this sodium, for example, we said there's more sodium outside the cell. If this cell was going to pump sodium, right, say there's 1 sodium inside the cell. If this cell was going to pump it out, it would have to use energy to do that because it's pushing it out to where there's already a ton. It's like pushing all of those little sodium together in one tiny little group, and that takes energy. You've got to work to make that happen. An example, or one of the most important examples of active transport that our cells utilize is called the sodium-potassium ATPase pump. Let me see. No, I was hoping I had an example of that to show you. The sodium-potassium ATPase pump. So let me tell you what that is. The sodium-potassium ATPase pump spends 1 ATP, okay, so 1 unit of energy, and with that 1 ATP, it pumps 3 sodium out and 2 potassium in to the cell. So think about our cell, right? Say I have a cell like this. And I told you guys in the beginning that it was really important for you to know where we have each of the ions. And I told you we have more potassium inside, right? Lots of potassium inside the cell. And we have lots of sodium outside the cell. Now that's the way that the cell is supposed to be at rest. This is the way the cell should be. Now diffusion, right, natural process tells us that these things are going to cross the membrane until they're even on each side. So if we just think about diffusion, diffusion says that sodium's going to diffuse in and potassium's going to diffuse out until they're even. But the cell doesn't want that. The cell wants there to be a difference, right? We'll talk about why later, like in the muscle chapter. But the cell needs to have more potassium inside and more sodium outside. So diffusion messes things all up for the cell. So the cell needs some way to fix that, right? To get rid of that and to move potassium back in and move sodium back out. The way that it does that is with the sodium-potassium ATPase pump. What that sodium-potassium ATPase pump does, again, is some of this potassium that leaked out here, right, and some of the sodium that leaked into the cell, it takes those ions and it puts them right back where they came from. So again, it takes 3 sodium and pumps them out at the same time that it takes 2 potassium and pumps them in. Now, since it's moving things up the concentration gradient, right, we said there's already more potassium inside the cell. So if it's putting even more potassium inside the cell, that's hard. That's going from low to high. Same thing with the sodium, right? There's already more sodium outside of the cell. So to take it and pump even more out there is hard. So that's why this is an active pump. That's why it uses ATP. Now there is something called secondary active transport. Now this secondary active transport is a passive process. It does not use any ATP itself. Okay, so the actual secondary active transporter, like for example, the sodium glucose transporter, does not use ATP itself. However, it relies on a pump that does use ATP. Okay, it relies on the gradient that's created by an active pump. So it wouldn't work if there wasn't a different pump that was active. Okay, but this pump itself does not actually use ATP. It is actually passive. That's why it's called secondary active transport. So this sodium glucose transporter, for example, if you look at the cell, right, I just told you guys that the sodium potassium pump pumps a bunch of sodium outside the cell. So we end up with a ton of sodium outside of our cell. And you guys know that diffusion naturally says that sodium is then going to want to enter, right? The only way we get it back out again is with the active pump that keeps pumping it out and pumping it out and pumping it out. But there is some sodium here that tries to leak its way back in. So what happens with this sodium glucose transporter is that this sodium just goes down its concentration gradient, right? It's just facilitated diffusion passing into the cell. And the glucose just kind of hitches a ride with it. So there's like a little transporter and the sodium, right, binds like it's going to come through. And when that happens, when the sodium is doing its facilitated diffusion, the glucose just sneaks along with it. So as the sodium comes down its concentration gradient, the glucose gets pumped into the cell as well. Another type of membrane transport is called vesicular transport. Now, vesicular transport is when we move materials into or out of the cell in vesicles. Hence, it's called vesicular, right? Vesicle. A vesicle is just like a little bubble of membrane. So, like if you have your plasma membrane like this, and a little bubble of it starts to like fold inward like that, and eventually it can like pinch off, and then this little bubble that you have, that's the vesicle. So it can come into the cell like this, or the membrane can start to fold outward like that, and eventually pinch off, and then you're left with a little vesicle. Just a little like bubble, a little chamber. So we can move things into the cell in vesicles or out of the cell via a vesicle. And sometimes the vesicle, like the little bubble itself, will just fuse with the membrane, and when it does, it'll dump its contents out. We'll see that as well. So the two major types of vesicular transport are endocytosis and exocytosis. So endo literally means within, and cyto means cell. So endocytosis literally is telling you within the cell. This is when we move things into the cell via a vesicle. So when the cell engulfs or brings in particles or fluid via a vesicle. And this is an active process. This uses ATP. Okay, this is not passive, it is active. Again, the plasma membrane, just like I drew right here, the plasma membrane forms a vesicle, right, a bubble around something, and that ends up pinching off, and then we have a vesicle inside the cell. So we brought something from out here into the cell, endocytosis. Now, there are actually two different types of endocytosis, depending on what it is that you're bringing into the cell. There is pinocytosis and phagocytosis. Okay, so both of these, pino and phago, those are both endocytosis because you're bringing things into the cell. It's just that we're being more specific when we say pinocytosis or phagocytosis. Pinocytosis literally means cell drinking. This is when the cell engulfs fluids. The way I remember that is Pinot. Pinot is like a liquid, right? A wine, like Pinot Noir, Pinot Grigio. Those are wines, liquids. So pinocytosis is bringing liquids into the cell in a vesicle. Phagocytosis literally means cell eating, right? A phage is eat. So this is when the cell engulfs objects in a vesicle. So for example, when an immune system cell takes in a bacteria, when there's bacteria infection in our body, our immune system cells will see the bacteria and they'll eat it. They'll take it in and then destroy it. Well, when that cell is engulfing that bacterium, right, when it takes it in a vesicle, that's an example of phagocytosis, right? It's taking in another cell, something that is solid. Exocytosis, right? Think X like exit, external, like outside. And then again, cyto is cell. So exocytosis is when materials are released from the cell when the vesicle fuses with the plasma membrane. So if there's something in this vesicle here, and then the vesicle comes and it fuses into the membrane and it dumps whatever's inside it outside. Again, this uses ATP. All of this vesicular transport is active. It all uses ATP. Now, things that can leave the cell via exocytosis include secretory products. So these are like things that the cell makes that it wants to secrete, something productive, that's like a hormone. Say the cell is making a hormone and it needs to release it. It can release it via exocytosis. Some waste products are also released this way. Okay, so here we see kind of both sides of this. Both of these things, both phagocytosis and exocytosis, this is all vesicular transport. So it's all active, meaning it all takes ATP. Now here, on this side, you see this is a bacterium, right? So streptococcus or staph or some sort of bacteria. That's bad. It's making the person sick, so their immune system cell is going to destroy it. So this bacterium gets taken into the cell via a vesicle. You'll notice that the membrane folds in like this and it ends up folding around it. And then you have the bacterium inside the cell in this little bubble. You don't want it just free floating around the cell because it can hurt the cell. So you keep it in this little vesicle, this little chamber. Now, since this was a solid object that was being brought into the cell, that is phagocytosis. Pinot would have been if it was just liquid. Remember, that is an example of endocytosis. In this case, there's a little lysosome. That was one of our organelles that had digestive enzymes that chew things up and break them down. The lysosome is going to fuse with the vesicle. The enzymes will break down that bacterium, and then we're going to release the waste products via exocytosis, right? Exiting the cell. You see this little vesicle or bubble just fuses with the membrane, right? It came from the membrane to begin with, and now it's going back into the cell membrane. But when it does that, it dumps the contents out of the cell. So since something is leaving the cell via the vesicle, that is exocytosis. Okay, so there are two kind of key processes that we're going to talk about to finish up this chapter: protein synthesis and cell division. We've kind of mentioned protein synthesis a few times throughout chapter 2 and 3, but we're just going to bring it all together now. Proteins are extremely important in the body, both structurally, right? Like collagen and keratin, they form a lot of our strong structure. And then also functionally, there are a lot of proteins that have their own specific functions, like hemoglobin that carries oxygen through our bloodstream. We have so many different proteins all over the body, and these proteins wear out. They don't last forever. So we constantly have to be making more proteins and more proteins. in order for the body to grow and develop, but also just in order to keep the body running so it doesn't break down. So protein synthesis happens all the time. Remember from chapter 2 that proteins consist of long chains of amino acids, and that those amino acids get linked together with something called a peptide bond, right? So we'll have like one amino acid and it gets linked to another, and then that gets linked to another. And we end up with a really long chain of amino acids. And that's like the very beginning of protein synthesis. Then remember that that chain gets folded or twisted, and and we end up making this really big structure. But it all starts with linking these amino acids together in the right order. Now, our DNA in the nucleus of the cell contains the coded instructions for how to make proteins. Hey, what these instructions are like a recipe, right? In a special code, and the code spells out the correct sequence of amino acids. So it says which amino acid has to go where. Okay, and proteins can be hundreds of amino acids long, and the sequence has to be perfect. So the DNA contains the instructions so that the ribosome, which remember is what actually makes this protein, so that the ribosome knows which amino acid is going to come next. When we look at our DNA, a triplet, right, which means 3, a triplet refers to 3 nucleotide bases. Remember the bases were like adenine, thymine, guanine, cytosine. 3 of those bases will code for one single amino acid. Okay, so when we're looking at our DNA, you know, say it's like A, T, C, G, C, G, right? These first 3, that's going to code for one amino acid. The next 3, that codes for one amino acid. So every 3 bases in the DNA is going to spell out one amino acid. Now, the process of protein synthesis. Okay, protein synthesis involves a couple major processes. The major processes are transcription and then translation. Okay, there's other stuff involved, but those 2 are the 2 major things that need to be done for protein synthesis. Really, first, before transcription, we do have gene activation. The gene is like the recipe. So there's the section of the DNA that codes for collagen, is like the gene that tells you how to make collagen. So we activate the certain part of DNA that we want to use. Say I want to make collagen. I need to go to my DNA, which remember the DNA is the whole darn cookbook, and I don't want to make everything. I just want to make that one recipe. I just want to make collagen. So you've got to flip through the cookbook and get to the right page, right? So go through the DNA and get to the right gene, and then you uncoil that section of DNA so that you can use it, right? That's gene activation. Essentially flipping through the cookbook and finding the correct recipe, getting that recipe out. Then you have the 2 major processes, transcription and then translation. Transcription happens inside the nucleus of the cell. This is when we make a copy of the recipe. Okay, so it copies the instructions for how to make the protein from DNA into a strand of mRNA or messenger RNA. Specifically, it's an enzyme called RNA polymerase that does this, that makes the messenger RNA. But I just want you guys to understand that transcription is when we copy the DNA to a strand of mRNA. Because remember, the DNA is not going to leave the nucleus, right? We're not going to take that master copy out of the nucleus. So we need some way to copy it so that we can take a copy out of the nucleus, out to our ribosomes. Then once we have the messenger RNA leaves the nucleus of the cell and it goes out into the cytoplasm where we actually make the protein. Out in the cytoplasm is where we have the next process, which is translation. Hey, this is when we translate from RNA to our actual polypeptide. In this case, it's the ribosome that does this. The ribosome reads the code from this messenger RNA. It literally latches onto the RNA and it goes down and it reads it codon by codon by codon, right? So triplet by triplet by triplet. The ribosome goes down and it reads the mRNA. And as it does that, it pulls together the amino acids and links the amino acids together in the right sequence. As the amino acids get hooked together, we get something called a polypeptide. That's just a chain of amino acids. Okay. And then after we have this big long chain of amino acids, then we just process that to make our final protein, right? So we use the rough endoplasmic reticulum for this. Sometimes we use the Golgi apparatus. They make some kind of final finishing touches, and we end up with a protein, right? So we started in the nucleus, and we went from DNA to RNA. Then the RNA leaves the nucleus and goes out into the cytoplasm to the ribosome. The ribosome reads the RNA and links together amino acids. Okay, we have our polypeptide. Then that polypeptide goes into the rough endoplasmic reticulum for processing. Sometimes it goes into the Golgi apparatus for processing. Then we have a protein. So we're going to kind of talk about transcription and translation in a little bit more detail. Transcription. Okay, this is again when we transcribe, right, a copy of DNA. So overall, we're going from DNA into messenger RNA. We're copying our recipe. The enzyme that was important, we said, was RNA polymerase. This is the enzyme that makes the copy of the DNA. It literally binds to a promoter sequence or a start sequence. So in your DNA, there's the actual recipe, but before that, there's like a little code that says, hey, start here, start here, start here, so that you know, here's one recipe, here's the next recipe, here's the next recipe. So there's little regions that say start, and then you have your gene, and then it'll say stop, so that the RNA polymerase knows exactly where that recipe starts and where that recipe ends. So the RNA polymerase will bind to this promoter region or this starting sequence, and then it's going to read the DNA code for that gene, right? It'll read the recipe. It hooks nucleotides together to form our messenger RNA. One thing to keep in mind is that when we're making the RNA, remember there's no thymine in RNA. So anywhere that we would put thymine, instead we put uracil. Then we have translation. Okay, we have our mRNA, and we're going to translate that into an actual protein or a polypeptide. This mRNA first leaves the nucleus through a little pore, right? Remember the nuclear envelope has pores in it, so things can get in and out. And the mRNA leaves the nucleus, and it goes to a ribosome that's out in the cell, in the cytoplasm. Again, these ribosomal subunits are going to latch onto the RNA, okay? And they're going to go down and read the RNA codon by codon by codon. Now, there is another type of RNA that's involved in this translation, and it's called transfer RNA or tRNA. This is what actually delivers the amino acids, brings them over to the mRNA and the ribosome so that they can be latched onto each other. I'm going to show you pictures, and it's going to bring it all together, I promise. So say I have my mRNA right here, and say my code is AUC GCC UCA, Okay, so say that's my mRNA. Remember I said each 3 bases is what we call a codon. So that AUC, that's a codon. GCC, that's another codon. UCA, that's another codon. So each triplet, each 3 bases on the mRNA is a codon. Remember, each of those is going to code for 1 amino acid. So like AUC, this means 1 amino acid. Now, the way that we get that amino acid is with the transfer RNA or tRNA. That's the one that brings the amino acid over. So what happens is for each mRNA codon, there's a tRNA anticodon that comes and binds to this. And each tRNA anticodon brings an amino acid. So if that was my mRNA down there, then the tRNA anticodons that match, remember A goes with T, but this is still RNA, so that means there's no T, it's got to be a U. U goes with A, C goes with G. So this up here is the anticodon that matches. Well, this anticodon, UAG, happens to have 1 amino acid attached to it. Then the next one comes. G goes with C, C goes with G, C goes with G. Right, so this is this anticodon. That happens to have another amino acid attached. So those will get connected to each other. And then you have the next codon down here, UCA. So U goes with A, C goes with G, A goes with U, right? This anticodon has another amino acid. Then those are going to get attached. So as we'll see the ribosome, but as the ribosome reads this and it moves down and down and down, it's going to read the next, there's to be a codon, and then an anticodon that matches brings an amino acid. And then it'll go to the next codon, find the matching anticodon, and then bring in another amino acid. And it just goes down the whole strand of mRNA, codon by codon by codon, so that it can string the amino acids together. Okay, so the overall summary of what happens in translation is that one mRNA codon translates to one tRNA anticodon, which translates to one amino acid. Okay, so the codon is down here, then the tRNA anticodon, which brings along an amino acid. All right, so first, remember up here in the nucleus, we have transcription. And transcription is where we take the DNA and we copy it to make a strand of mRNA. Then this mRNA leaves the nucleus through a little pore and it goes out into the cytoplasm. And at the cytoplasm, we're going to have a ribosome, right? There's a small unit up here, and then we'll see another unit on the bottom is going to come, a large unit, and they're going to snap onto this mRNA. Okay, so this strand, that's the mRNA. That's like the recipe that's going to tell us how to make this protein. Down here, each 3 bases, again, is a codon. So there's one, right? Each 3 of these is a codon. The tRNA, right? This is the tRNA with its amino acid attached. And the 3 bases here on the tRNA, those are the anticodons. So you'll see they go together. Again, A with U, U with A, C, G with C. And that's got its own specific amino acid. So everywhere that the tRNA says UAC, it's gonna mean this amino acid. So you see here's the first amino acid, right? And then the next, here's the tRNA with the amino acid attached, and that's gonna come into the second site. Now we'll see that there's going to be enzymes that are going to come and link these two amino acids together. And then this ribosome is going to move down. Okay, so now you see that happening. We linked these amino acids together with a peptide bond. The ribosome moved down to the next part of the mRNA. This tRNA is now done. We've taken the amino acid off of it, right? So we kick this out and it goes and gets another amino acid, it gets recycled. Now, since the ribosome moved down, it's got another open binding site. So the next tRNA anticodon will come in and then we'll form another bond between those amino acids. And then the ribosome will move down again, right? And this one will get kicked out and recycled. And that just keeps going on and on and on until we've linked together hundreds of these amino acids. Okay, that is protein synthesis. I realize it is really complex. I try to simplify it as much as possible by not talking about the names of all of the enzymes and all of that. So hopefully you're able to kind of grasp the overall process of what's happening and what's important in the process. The last major thing that we're going to talk about is cell division and the process of cell division, the stages. What cell division is literally when one cell divides to make two cells. This is how our cells replicate. Ultimately, a person starts as one cell. And in the mother, in the uterus, that cell divides and divides and divides and divides, and then you end up with trillions of cells that make the person. Our cells continue to divide, though. That's how we grow, and then that's how we repair our tissues, is cells dividing. So cell division is very important. There are two major types of cell division: meiosis and mitosis. Meiosis is really, it's not a very common cell division. This is the cell division that happens only in the ova or egg and the sperm. So meiosis only happens in our reproductive cells. This is the cell division that makes the egg in the woman and the sperm in the man. Now this cell division is different. It's like two divisions. The cell divides once and then it divides again. So because it divides twice, it ends up producing 4 daughter cells, right? Like 4 new cells. And each of those cells only has half the number of chromosomes as the parent. So half the genetic material. Most cells have a total of 46 chromosomes. We have 23 pairs, right? So 2 of this chromosome, 2 of this one, 2 of this one, 2 of this one, right? All the way down, all 23 chromosomes for a total of 46. The egg and the sperm don't. The egg and the sperm only have 1 of each chromosome. So they only have 23 chromosomes each. Now, in order to be a living functional being, you need a full set of 46 chromosomes, but the egg and the sperm don't, right? So think about why. Why is it that the egg and the sperm only need half the genetic material? Well, the reason for that is that their entire purpose is to come together with the other one, right? The sperm and the egg come together to make a new being. And each of them is going to bring half of the genetic material of the new being. Right? So the egg is going to bring 1 of each of the chromosomes, and then the sperm is going to bring 1 of each of the chromosomes, and when they fuse together, they make a full set. Right? So that's why you get half of your genes from your mom, half of your genes from your dad, they come together, and then all of the new cells that come from that are the new being. They all have 46 chromosomes. So that's meiosis, right? It happens in the egg and the sperm. It produces 4 cells instead of 2, and each of the cells has only 23 chromosomes. That's all we're going to say about meiosis. Mitosis we'll look at in more detail. Mitosis is the normal cell division that occurs throughout the body. This is how our body grows. This is how we repair our tissues. This mitosis is happening all the time, all over your body. Excuse me, I get the hiccups. Oh, goodness. Okay. In mitosis, it's a cell division that produces only two daughter cells. And each cell has the same number of chromosomes as the parent. So one cell divides and it produces two identical cells. They look exactly like the mother cell, exactly like the first cell. And they all have the same 46 chromosomes. Now, when we look at a typical cell, most cells do not spend their entire life dividing. Most cells only divide at very rare, very specific times of their life cycle. Most of a cell's life cycle is actually spent in interphase. This is not dividing. This is the time when the cell's just doing whatever that cell does, right? Depending on what kind of cell it is and what area of the body. Once a cell goes into the dividing stage, right? Once it leaves interphase, then it's going to actually divide. It divides in three key stages. Somatic, that just means body cells. So not the sex cells, not the egg and the sperm, all of our other body cells divide in three stages. First is DNA replication. This is when the cell makes exact copies of its chromosomes. Because remember, we said one cell is going to divide to make two cells that are identical to the first cell. So each cell is going to have a full set of chromosomes. That means that the first cell has to have two copies, right? That first cell normally has its typical 46 chromosomes, but if it's going to give 46 to this baby and 46 to this baby, well, it's got to double up sometime. So that's what happens first. Right? The cell that's going to divide duplicates its DNA so that it has two copies. Once the cell has two copies of its DNA, then it does the actual process of mitosis. Hey, mitosis is how the cell divides the new genetic material equally so that each baby cell gets 46 chromosomes. And then finally, so we have DNA replication, mitosis, and then the very final thing that happens is cytokinesis. Cytokinesis is the actual physical splitting of the cells, right? So the cytoplasm and the organelles and all the other stuff that's in there just physically gets separated or pulled apart into the two daughter cells. And cytokinesis, the separation, actually starts during mitosis. And then once the genetic material is completely divided in mitosis, the cytokinesis just has to finish, kind of pinching off into two separate cells. Okay, so the cell's life cycle. Again, all of this is when the cell is not dividing, right? So most of the time the cell is in interphase when it is not dividing. The actual division of the genetic material, which is mitosis, you see here, and then cytokinesis, which is the physical splitting of the cell, I said starts during mitosis and then continues. I do want you guys to be able to name these phases in order, right? So interphase, prophase, metaphase, anaphase, telophase. The saying, the acronym I use to remember that is I picked my apples today. I picked my apples today. I for interphase, P for prophase, M for metaphase, A for anaphase, T for telophase. Okay, so you should be able to put them in order. And then you should also be able to tell me what's happening in general in each stage. And that's what we're going to talk about now. So during interphase, this is just the cell normally being the cell. During interphase, the DNA is loosely kind of messy, right? You can't see nice tight chromosomes. The DNA exists as chromatin, right? So when you look at the cell, you see just messy, loose DNA. And then you see the centrioles right over here, right next to each other, just outside the nucleus. Just before the cell is going to divide, go into mitosis, remember the DNA duplicates so that we end up having two pairs or two sets, and the DNA gets really like nicely, tightly organized and coiled into chromosomes, which we will see in a second. Okay, so we'll go through each of the phases of mitosis, starting with prophase. So we already, remember right before mitosis, it already duplicated the DNA so that there were two pairs. In prophase, we see that the genetic material, right, the DNA gets really organized. It gets really tightly coiled like this, and we see these nice chromosomes start to appear. Nice, like kind of tight rods like this, and you see something called a centromere in the middle. Okay, so we see that happening. We see chromosomes form during prophase. We also see that the centriole pairs move to opposite sides of the cell, and they stretch their microtubules between them. So they form the spindle apparatus, and then the nuclear envelope disappears. So we start to see chromosomes form, right? If this is the cell, we see the centriole pairs start to move to opposite sides and stretch these spindle fibers between them. So you'll see this thing kind of like radiating out like this, and the nuclear envelope disappears. So you don't see that distinct nucleus anymore. Then during metaphase, the chromosomes all align in the center of the cell. So we'll see, like if this is the cell here in the middle, the chromosomes will all line up nicely across the middle like this. The spindle fibers, right, from the centrioles will attach to the center of each of these, to the centromere that's in the middle. So you'll see the fibers all attached like this. So during metaphase, you'll see this nice thick metaphase plate or line in the middle. During anaphase, the centromeres divide and the spindle fibers pull the chromatids. They pull the DNA apart. So what you end up seeing is you'll have your little centriole pair here and these spindle fibers, and they're pulling half of the DNA this way, and then on the other side you see centrioles attached to the DNA, and they're pulling the DNA this way so that it's now split. And then during telophase, you see new nuclear membranes form. So over on this side, a new nuclear membrane, and then over here, another new nuclear membrane. And then the chromosomes uncoil again. Finally, we said the last thing in cell division is cytokinesis, and that's literally just the physical division of the cytoplasm. So taking the cell membranes, and it just starts to kind of pinch in, right? So we'll have one cell, and then it just starts to kind of pinch like this, and then that pinch goes further and further and further and further until it's two separate cells. So here you guys can see the whole process. Up top here is a drawing, but then on the bottom, these are actual pictures. So these are taken with a microscope of real cells that are dividing so you can kind of see what happens. Both of these are showing prophase. Remember, during prophase, the centrioles will move to opposite sides and they'll start to stretch these fibers between them. And then the DNA starts to get really tightly coiled. So you can see like how dense these little rods are starting to form here, right? And here you can see the same thing, and the centrioles moving across and connecting their rays to each other, their microtubules to each other. You also see that the nuclear envelope disappears. During metaphase, that's pretty easy to pick because you get this this perfect dense dark line of chromosomes in the middle. Right, so the metaphase plate is where these chromosomes all align. You'll notice that there are the centrioles on either side, and they've got these fibers that are attached to the DNA on either side. During anaphase, the DNA splits, right? You'll notice that they're no longer attached to each other, and the centrioles are pulling their DNA over to each side, right? So you can see it here, that split. And then during telophase, you have new nuclear envelopes that have formed, and the DNA is uncoiled. And then finally, cytokinesis is the final split, once it's completely split. And now you'll notice that we have two completely separate cells. So you guys should be able to tell me the stages in order, and you should be able to tell me a little something about what happens in each of the stages. And I normally will bold the things that I think are important from each stage, so you can typically just stick with that. Okay? All right, guys, so tomorrow is just supposed to be a review day for you guys. So what I'm going to do is I'm going to post a file that has like all of the PowerPoints, but then everything that I want you to focus on for the exam, I've got highlighted in a red. So you can look through that file and it should really focus you for your last couple days of studying. And then I'll also post some practice questions. So after you've gotten some studying in from the review file, go ahead and work through the practice questions. The practice questions are not everything that you have to know. It's just a way to kind of go through and get a little bit of practice going through and answering questions. So make sure you're really comfortable with all of those practice questions. So I won't have a video like this for tomorrow. Tomorrow is just a review day on your own with the stuff that I'm posting on Canvas, and I'll post it right now. And then the next day, so Thursday, is you guys' first test. Okay, so Thursday will be you guys' first exam. If you guys have any questions today or tomorrow, please feel free to shoot me an email.